Breaking news! New research provides insight into the synthesis of gold nanoparticles with controlled shapes via manipulation of the type and concentration of halide and silver ions. Different metal nanoparticle shapes can be attained by varying synthetic conditions, since the shape, or morphology, of nanoparticles determines how molecules can bind at its surface. Nanoparticles can be leveraged to selectively catalyze reactions. This has relevance in fields such as biological labeling, optics, spectroscopy, and catalysis. Therefore, research in the area of controlling the nanoparticle shape is important to maximize their utility, since their physical properties depend on their shape. Nanoparticles are commonly produced through colloidal or solution-based seed-mediated synthesis. The seeds consist of a metal center and serve as a nucleation site or origin of the crystal nanoparticle structure in a bottom-up approach, meaning that the nanoparticles are atomically assembled and grow over time. A key topic is silver under potential deposition, a phenomenon in which a metal ion is deposited on an existing metal surface at a potential that is more favorable than the potential required for bulk deposition, resulting in the formation of a monolayer that can stabilize surface facets of differing energies depending on the ratio of metal ion to neutral metal. Surface facets are the consequence of a specific packing order of ligands. This is described by a bracketed notation that describes the orientation of dominant exposed faces along a particular crystallographic plane. This allows for silver to deposit onto certain sites of a gold nanoparticle surface, which influences the growth rate and morphology of the nanoparticle.
Remember that the two key components of the seed-mediated growth mechanism are the reduction of aqueous gold cations to solid gold particles and the binding of those particles to the gold seed, which grow the seed to a nanoparticle. How do halides affect the growth rate? Firstly, the gold ions coordinate with the halides to form complexes which are less soluble in the growth solution with increasing halide size. As such, it decreases the concentration of gold ions, thereby decreasing the rate. These complexes also reduce the reduction potential of the gold ion, thus decreasing the cell potential of the overall redox reaction and thereby making the reaction less favorable due to the direct proportionality of Gibbs free energy to negative cell potential. The reduction potential of the gold ion decreases as the size of the halide it coordinates to increases, so growth rate also decreases as halide size increases. Thirdly, the halide anions themselves can inhibit the rate of nanoparticle growth by binding to the surface of the gold nanoparticle, thus blocking the reduced gold particles from growing the seed. These gold halide bonds must be broken before the gold particles can attach, and the strength of the gold halide bond increases with halide size due to the hard-soft acid-base chemistry.
The trend of halide absorptivity on gold surfaces can be understood through the consideration of hard-soft acid-base theory. Hard means less polarizable due to a smaller size, and soft means more polarizable due to a larger size. Since the gold metal center can be classified as a soft acid, a softer base will correspond to a larger halide, as it is more favorable for soft acids to interact with soft bases, because smaller halides have less competition to bind to the surface of the gold nanoparticle.
We expect larger halides to reduce the growth rate of the nanoparticles, resulting in lower index facet morphologies. But how do we test this? There are two main ways to add halides to growth solutions. One is through adding a halide-containing surfactant, and one is through adding halide anions by using a salt. The researchers first looked at the difference between chloride and bromide by using their respective surfactants. The CTACl growth solution had a 2-to-1 reducing agent to gold ion ratio, whereas the CTABr solution had 16-to-1 ratio. Based on previous research, we expect that the 16-to-1 ratio would have the fastest growth rate, as increasing the concentration of reducing agent increases the rate of reduction. So we would expect the CTABr growth solution to have higher index facet morphologies. However, chloride solutions still had a faster growth rate than the bromide. To look at the difference between bromide and iodide, the researchers first looked at the effect of bromide ions. In order to do so, two identical growth solutions were prepared, but 5 millimolar sodium bromide was added to one of them.
In the absence of bromide, the nanoparticles take on a tris octahedral shape, indicating a rapid reduction rate in stable particle growth. When bromide is introduced at a concentration of 5 micromoles, the nanoparticles become cubic shaped, showing that bromide ions acting as a soft base have influenced the growth rate by stabilizing certain facets and reducing the reduction rate. This figure supports the conclusion that halides, like bromide, can control the growth rate and shape of nanoparticles by altering the reduction kinetics.
An identical procedure was used with sodium iodide. However, the amount of iodide necessary to result in similar or greater decreases in rate is 2 magnitudes less than the amount of bromide needed. This relationship also supports the hypothesis that larger halides hinder the rate more significantly than smaller halides.
The authors outlined design considerations that enable control over nanoparticle morphology. First, increasing the concentration of ascorbic acid accelerates the rate of gold reduction through kinetic control. Second, larger halides slow growth by decreasing the reduction potential. Third, in the absence of larger halides, higher silver ion concentrations can stabilize less energetically favorable morphologies through underpotential deposition. Fourth, adding trace amounts of halides either stabilizes or destabilizes the underpotential deposition layer, depending on the size of the halide, meaning that higher energy surface facets can be stabilized with less silver when the silver underpotential deposition layer is destabilized. Fifth, high concentrations of larger halides hinder silver deposition, limiting accessible morphologies. Lastly, kinetic trapping due to the underpotential deposition layer and chloride surfactant enables access to a broader range of morphologies. The authors conclude that these insights not only explain the formation of various gold nanoparticle shapes, but also extend to other nanoparticle syntheses, suggesting a universal framework for controlling nanoparticle design, which paves the way for precise catalytic applications and further research involving multiple additives during nanoparticle synthesis. The authors conclude the same effects of trace halides in the presence of silver ions in seed-mediated colloidal synthesis, as were studied on electrode surfaces in electrochemical cells in this paper. Research by Xiao has involved sulfide ligands in halide-free seed-mediated synthesis. Sulfide controls reduction kinetics but also absorbs onto facets, reducing gold nanoparticle thickness. Research by Wolf and colleagues further explores halide-free synthesis and shows that nanoparticle size is related to a lower ratio of the surfactant to silver ions. Subsequent papers have continued to address multiple additives in the colloidal synthesis of gold nanoparticles. Halford observed deviations in expected nanoparticle shape when different bromide surfactant product lines were used to make palladium tetrahexahedral nanoparticles. Morphologies were corrected by adding known volumes of organic solvent back to their surfactant after evaporating impurities. This raises questions on the effect of solvents, suggesting that micelle formation impacts the reaction rates of nanoparticle growth.